My name is Geoff Chong. I'm a lymphoma oncologist from Melbourne, Australia. I'm an associate professor at the Olivia Newton-John Cancer Centre in Austin Health, and my broad research focus is immunotherapy for lymphoma, particularly in early line therapies, actually. So with regards to this trial at this AACR meeting, we presented the ELM2 trial, particularly a follicular lymphoma cohort. So in this trial, we studied odronextamab, which is a CD20 CD3 bispecific antibody in a cohort of follicular lymphoma patients who are relapsed refractory. They need to have received already at least 2 lines of therapy. Patients were heavily pretreated. Median prior lines was 3, and nearly half were POD 24. So this abstract was a primary analysis where all 128 patients had at least 52 weeks of follow-up. The primary endpoint was objective response rate, and this was 80.5% with a CR rate of 73.4%. The median PFS was 20.7 months. The safety was generally manageable. Halfway through the study, the step-up dosing was revised to a new schedule. So with the 0.7 to 4 to 20 milligram step-up dosing in the first cycle, CRS was very well managed. It did occur in 56.7% of patients, was grade 1 in 45%, grade 2 in 10%, and only 1 patient had grade 3 CRS. The target dose was 80 milligrams weekly, which then went to 160 milligrams 2 weekly, and then for patients in CR after 9 months, went to 160 milligrams 4 weekly. So the other major toxicity to note was infection, which did occur in 79.7% of patients. COVID-19 accounted for 36.7% of those. Certainly we saw high response rates and manageable safety profile in this study. This has led to a phase 3 program, so-called Olympia studies. There are 3 studies which involve follicular lymphoma. So the first being Olympia 1, which is a head-to-head phase 3 study comparing odronextamab to investigator choice chemoimmunotherapy such as R-CHOP or R-bendamustine in patients with untreated follicular lymphoma. Both arms received maintenance. So it's either odronextamab in the odronextamab arm or rituximab in the chemoimmunotherapy arm. Primary endpoint is CR at 30 months. So this trial will give some indication of the efficacy and safety of odronextamab against current standard of care chemotherapy-based treatment. Olympia 2 is a phase 3 randomized trial comparing the addition of odronextamab to standard chemotherapy, such as CHOP or CBP, again in patients with untreated follicular lymphoma. In this study, all patients received chemoimmunotherapy. So there are 3 arms. There's chemotherapy plus odronextamab with no maintenance. There's chemotherapy plus odronextamab with maintenance odro. Or the standard arm is chemotherapy plus rituximab and then maintenance rituximab. Primary endpoint again is CR at 30 months. This trial will determine whether the safety and efficacy of standard immunochemotherapy can be improved by replacing rituximab with odronextamab. Olympia 5 is a phase 3 randomized trial for patients with relapsed or refractory follicular or marginal zone lymphoma. So here the randomization is to either odronextamab plus lenalidomide or standard R-squared with rituximab plus lenalidomide. And the primary endpoint here is PFS. So this can determine which chemo-free option is better for relapsed refractory patients. Yes, look, I do have some general comments about the current bispecifics. There's certainly high response and complete response rates in the relapsed and refractory population. Clearly, we need to see if this will translate into the first-line population. The PFS and duration of response need more follow-up time, obviously. CRS and ICANS are very well controlled with the step-up dosing regimens and pre-medication. I think one challenge is the significant infection rate. Really, we see this all the bispecifics, especially with extended dosing. So I think it's good to move towards fixed duration therapies. As you said, going forward, I think we need to define the optimal setting for the use of bispecific therapies. Should we be using them first line? Should we be using them in the relapsed refractory setting? Can we use them sequentially? Optimal combinations, such as with chemotherapy or biologics, novel agents, targeted therapies, and then clearly risk stratification, patient selection, trying to find biomarkers for response. These are, I think, the significant questions we have going forward with bispecifics for follicular lymphoma. I think this is a good question. Clearly we're manipulating the immune system here with the bispecific approach. And remembering, as you say, there are CAR-T and there's numerous other approaches. There are other immune cell engagers, for instance, not just the T cells. I think it will be important to determine which setting to use these treatments in, particularly with respect to CAR-T. Is it better prior to CAR-T? Can we use them post CAR-T? Can you use sequential bispecific, either the same bispecific or an alternative one? Can you engage the different immune cell types in sequence? There are all these questions, and they need to be investigated. This will take time. Clearly, but I think we're certainly on to a good treatment for the future, and certainly this trial provides a good setting for the further investigation of odronextamab.